Good morning, everyone. Today, we are going to start with our first lesson on biomedical sciences. As you can see, this is just an introductory lesson as well as the lesson on blood typing. Okay, so throughout these next 2 weeks, we are going to have recorded lessons. Okay, so this is the first recorded lesson for biomedical sciences. My name is Mr. Hakim, and I will be going through with you this module for the entire 4 weeks that we are going to be together. Now, these are the things that we are going to cover for this lesson. So the first part is what is biology and biomedical sciences. This is introduction. Then the second section will be on the constituents of blood. And finally, we are going to go through the section of blood group typing. In other words, how do we differentiate between the different blood groups? Now, what is biology? Now, biology itself is actually a word that we took from the Greek word, which is bio and logos, okay, which is bio meaning of life, logos the study, and so biology means the study of life. Now, biology actually encompasses everything that is living on this earth, so that it can be humans, it can be animals, it can be birds, fish, anything that is considered to be living things all fall under the purview of biology. Okay, but however, our module today is on biomedical sciences. So biomedical sciences is a very specific branch of biology. So biology is the bigger picture and biomedical sciences is very specific. Okay, now what exactly what exactly what exactly are biomedical sciences? So you can see here, it is the primary focus on biology of human health and disease. Okay, so in other words, we are focusing on humans, right? And how the body works. Right? We don't care so much about animals. We don't care so much about fish. We don't care so much about birds, so on and so forth. We are focusing primarily on humans. Now, from the word medicine, of course, we know that it also has to do with disease and how we come up with treatments for these diseases. Okay, so what are the things or what are the sciences that are involved in these biomedical sciences? You will get things such as your anatomy, which is your study of the body, of body parts, right? Then we have physiology, basically how the body works. So this is more functional rather than just studying what the body parts do, right? Then we have cell biology, basically studying the cell. Then we have biochemistry, talking about the chemical processes here, right? And then we have microbiology. This is focusing on microorganisms. Genetics, focusing on DNA. Molecular biology, focusing on biological molecules. Immunology, your immune system. And bioinformatics, this has to do with the statistical analysis, right, of biological functions. Right now, this bioinformatics is actually a very advanced kind of mathematics, which is dealing very specifically with with the function of the body. So this is something that we won't go through, okay, thankfully. So we will be focusing more on the other different types of sciences in this module. Okay, now biomedical sciences are the major focus of bioscience research and funding in the 21st century. Now, this is very true, especially in Singapore, because Singapore itself has a very strong biomedical research hub. Okay, there are many well-known how should I say this, biomedical companies that are known internationally that produce drugs, that produce medicine, right? which have specific research labs as well as manufacturing plants in Singapore. So we are actually a biomedical research hub. Okay, so if you are thinking of a career in the biomedical sciences, it is something that is very strong in Singapore, and I think it will be continually be relevant as even when you start to go to work. Okay, now what are the different divisions of biomedical sciences? So all the different things that biomedical sciences encompass, the different things that are being studied, can be categorized under three different categories. Okay, so these three categories are life sciences, physiological sciences, and medical physics or bioengineering. Now, life sciences focuses mostly on very specialized and micro studies. What do we mean by this? So, life sciences deals mostly with things such as your um, study of the cells, study of microorganisms, and basically how they work, right? So this is very specialized, and they are very micro studies dealing with very specific cell structures or tissue structures. Okay. Now for physiological sciences, it is also specialized, but it is different from life sciences in a sense that it focuses more on the processes as well as as well as the diseases that can come from the there's something wrong that happens in the process, which our body goes through. If there's anything wrong with it, there will be diseases that arise from it, right? So physical sciences deals mostly with that. And finally, we have medical physics. Now, medical physics or bioengineering has to do with the physical processes that are used in biology. Okay. So things such as your how do we make machines? Okay, these are all actually under engineering that help to diagnose specific patients and what disease they have. All these machines fall under bioengineering and medical physics. So these are some examples. Okay, these are not just the only three things that life sciences study. Of course, there are much more. Okay, but these are the three things, just three examples in which fall under life sciences. So hematology, which is the study of the blood, right? And this is something that we are going to go through today. Microbiology, which is the study of your microorganisms, such as your bacteria, your fungi, viruses, all these fall under microbiology. And finally, cell biology, which is the study of the cell. Then we have physiological sciences. So here, like I've mentioned before, we talk mostly about the functional processes of very specific organs or organ systems, right? When we learn how they work, we also learn when there's a failure in how they work, it will arise in some diseases, right? So cardiac physiology, as you can tell, it is the study of how the heart works, right? Neurophysiology, which is the focuses on the brain as well as your nerves, right? All of us have nerves that the brain that stem from the brain as well as your spine that conducts electrical signals for you to be able to move and to coordinate your movements, so on and so forth. And finally, gastrointestinal physiology. Of course, there are much more things that we study, but these are just the three examples that I've decided to give to you. Okay, so gastrointestinal physiology is the study of how your digestive system works. And finally, nuclear medicine falls under medical physics and bioengineering as well as clinical engineering. Now, nuclear medicine is actually quite a broad sorry, nuclear medicine is actually quite a broad topic of study, because if you think about it, nuclear medicine not only deals with specific drugs, which is the medicine that you are given, but it also deals with specific radiation therapy. Now, all of you here should be familiar with what cancer is, right? So cancer is basically the overgrowth of cells, the unneeded overgrowth of cells, right? That leads to specific complications and diseases in the body, right? And one of the key things in which we use to combat cancer is radiation therapy, right? But radiation therapy deals with specific radioactive waves, right? And this has to do with actually physics. So we are using actually physics to as a treatment, right? As the knowledge of our physics helps with the treatment of specific cancers. Okay, not all cancers, but specific cancers. So this is how we use other sciences, okay, and other knowledge to go and help complement biology as well as biomedical sciences in the treatment of diseases in patients. Okay, then clinical engineering is basically how we make, right, the science of making your medical apparatus, right? So here you can see this is a I think this is the it should be the blood pressure machine. Okay, so they are making a blood pressure machine here. So the concept of blood pressure falls under physiological processes, but how we make the apparatus, okay, or the machine needed to measure blood pressure falls under engineering. Okay, so now that we have gone through a very brief introduction on what biology is and what biomedical sciences is, or biomedical sciences are, so we will now move on to the next section, which are the constituents of blood. Okay, because blood is one of the main topics that we are going to go through for this entire module, and there will even be an experiment that we'll be conducting once we come for the third lesson, right? So you might have already learned this in your primary school or even Sec 1 sciences. Okay, so I'm going to go through specific concepts that you may be familiar with, but there might be certain things that I'll be going in depth, okay, because we are trying to push you to learn more. So there are some concepts that I'm going to go through which might be a bit advanced. Okay, so I'll go through this section quite slowly. Now, what does blood contain? Now, blood is considered a fluid tissue. Now, tissues are basically what we call a collection of cells, right? Now, why do we call blood a fluid tissue? Because if you think about a collection of cells in our body, such as your skin, such as your organs, all these are actually made up of tissues, right? And we always think about them as something that is solid, but blood is special. Okay, blood is fluid, right? It's almost liquid-like, right? That's why it's called a fluid tissue. But not only is it a fluid tissue, it's also considered a fluid connective tissue. Now, why is it connective? Because it connects, if you think about it, all organs together. Now, what do we mean by connects all organs together is that blood can travel, or blood is being pumped by your heart to all parts of your body. So there is no part of your body that has no blood, right? Even your eye has blood, your brain has blood, your skin has blood. Every single part of your body, of course, except for your hair, has blood, right? Hence, your blood connects all your organs together because your blood can flow to all parts of your body, right? Now, these are the main constituents of blood, and they are made up of plasma, red blood cells, white blood cells, and platelets. Oops. Okay, give me a moment. Okay. Okay. Right. Now, so these are the 4 things. Okay, apologies for that. These are the 4 constituents of your blood, right? We have plasma, red blood cells, white blood cells, and platelets. And all of these are either cells or they are not cells. Okay, but I want you to first, before I go any further, right, is to tell you which are the cells and which are not the cells. Now, red blood cells, as you can tell, of course, is a cell. White blood cells are cells as well. Platelets are fragments of cells, so they are not really cells themselves. Okay, they are just fragments of them, and they have their specific purpose, right? And plasma is actually just a kind of fluid. Okay, it's not a cell. So if you can think about it, right? Plasma and platelets are not really cells, are definitely not cells. Red blood cells and white blood cells are definitely cells. Okay. Now, if you were to collect your blood into one specific test tube, you are going to get blood which looks like this, but we can actually separate your cells into via centrifugation. Now, centrifugation is basically a process, right, in which we use the molecular weight, okay, or we should I say we use the mass of your specific constituents, okay, using mass to separate, right? So what we will do is that the centrifugation process involves spinning the blood in a machine, right? And when it spins the blood, right, there are forces acting on the blood that pulls the heavier part of your blood to the bottom of the test tube, right? While the lighter parts of your blood is left at the top, right? So what you are going to get here is something like this, right? After the centrifugation process. Now, from here, you can tell the top here is your plasma, right? And this is 55% of your blood. So if you think about it, a majority of your blood is actually just fluid, right? It is just the plasma. Then we have this white middle section here is so thin here, right? The white middle section here is your white blood cells and platelets, okay? And finally, we have your red blood cells, okay? So your red blood cells actually account to around approximately 40, if I'm not wrong, it should be around 40. Now, so we have around 40% of your red blood cells. 40% of your blood are red blood cells. Okay, so that would mean that your white blood cells and platelets are around 3 to 5%. Okay, now plasma, which is around already, remember, is around 55% of your blood. Okay, is made of 90% water, in which the following are dissolved in. So these are the things that are found inside your plasma. Okay, so proteins such as fibrinogen, prothrombin, and antibodies. Okay, now what fibrinogen and prothrombin are? Okay, these are what we call blood clotting. Blood clotting proteins. So these proteins are actually involved in your blood clotting process, how your blood clots. Okay, and finally, antibodies. Now, antibodies, we will go through them very briefly later, but basically they are proteins in which we use to combat pathogens or foreign microorganisms that has entered our body. So things such as your bacteria or fungi, or even viruses that have entered our body and causing harm. These antibodies are there to neutralize these pathogens. Then we have salts such as chlorides and sulfates of calcium and sodium. So these are just basically salts that are needed for our body to function properly. And we have food substances such as glucose, amino acids, as well as fats. Okay. Now we also the blood also, which is which has mostly the plasma, also carries waste products such as your urea, uric acid, and creatinine. Okay, now creatinine is basically a waste. I think you all should know what urea and uric acid, all this, urea especially, this is a main constituent of your urine, right? Okay, your urine will have urea, which is being removed via the kidneys. Okay, then creatinine, okay, is a waste product that is being produced when muscle breaks down. Okay, when your muscle breaks down from whatever process, whether it's through exercise, whether it's through old age, okay, it produces a lot of it produces this protein called creatinine. Okay, now this creatinine, okay, it is it is also being removed via your kidneys. Okay, so but if there is an issue with your kidney, right, your creatinine level in the blood will start to rise. So actually, this is an indicator. Okay, this is just some additional information for you. It's an indicator of kidney function. Okay, because if your kidney is functioning properly, okay, your creatinine levels are going to be quite low because your kidney can actually filter the creatinine from your blood. But if your kidney is not functioning, you are going to see a very high level of creatinine inside the blood. Okay, as well as hormones. Okay, now hormones, I think you all should know, are just basically um, proteins, okay, that help with many different functions inside or processes inside your body, such as growth, development, okay, things such as your puberty. As you go through puberty, you know that the male hormone or the female hormone, okay, plays a very large role in how your body develops. Okay. Now, so the next constituent of blood, other than your plasma, is going to be your red blood cells, or another word for them will be erythrocytes. Okay, this is just another word for your red blood cells. Okay, now they constitute 99% of the cells. Okay, 99% of the cells. So if your blood is made up of white blood cells and red blood cells, your red blood cells make up 99% of all the cells. Okay, they are produced in the bone marrow. Okay, now the bone marrow is found in very specific parts in your body, but one of the main parts that is being found is actually inside your spine, right? Okay, it's found in the spine. Okay, now what is it responsible for? The bone marrow is actually responsible to go and make all your blood cells, right? To make, oops, blood cells. Okay, now red blood cells, after they are being made and after they do their job, they are destroyed at the spleen. Okay, so the spleen is basically an organ that is found very near your liver. Okay, you will learn more about the spleen when you go to upper sec lah, but for now you don't really need to know too much about it. Yeah, so once it is being used, okay, so your red blood cells can last for around 3 to 4 months. Okay, so like I said, the lifespan is around 120 days, so this is around 4 months. Okay, so after 4 months, right, the old red blood cells who are around 4 months, okay, they are already used up for very long. Okay, so you want to recycle the red blood cells. What we will do is we will bring them to the spleen to destroy them, and once we get the constituents, we are going to use that material to make new red blood cells. Okay, so this is the bone marrow, as I've mentioned. Okay, produces red blood cells and destroyed in the spleen. Okay, now what are the function of your red blood cells? Okay, is that they transport oxygen to cells in the body. Okay, and this is just basically the main function of red blood cells. This is their main function. Okay, which is to transport oxygen to all the cells in your body. Okay, so how is the red blood cell adapted for its function? Okay, you are going to get a red blood cell that looks like this. Okay, it's filled with a red pigment. Okay, it has hemoglobin. Okay, so this red pigment of hemoglobin is very important because it actually is the one that binds and transports oxygen. Okay, without hemoglobin, your red blood cells cannot function. So this is actually very important, right? Without the hemoglobin pigment found in the red blood cell, okay, you cannot transport oxygen at all. Okay, and if you realize red blood cells also has no nucleus. Okay, now why is it important that it has no nucleus? Okay, is that it will allow you with more space lah, okay, to go and carry hemoglobin into the cell, and hence when you have more hemoglobin, this leads to more oxygen that you can carry, right? And finally, it has surface proteins. Now this part, okay, is actually very important as well, because this has to, or this is very related to blood typing, which we are going to learn later. Okay, so these surface proteins, right, basically uh, form recognition markers, okay, as to how you can identify specific blood types. Okay, this one we will go through later on. Okay, you can tell that the blood also has some form of biconcave shape, okay, and increases surface area to volume ratio for efficient uptake of oxygen. Okay, now what do we mean by increased surface area to volume ratio? This one, right, basically you can think of it this way. The smaller I am, right, the smaller I am, okay, but if I have a large surface area, okay, I can be something small, right, like something like this red blood cell here. It is small in volume. Oh my goodness. Okay, apologies for that. Okay, now where were we? Yes. So increases surface area to volume ratio. So basically, we can think of surface area to volume ratio as something as if you are small in volume but large in surface area, this will make me more able to absorb more oxygen. Okay, now why this is so is because, okay, okay, apologies for that. think there was something wrong there. Okay, yeah. So right, it's able to absorb more oxygen, right? So when it's able to absorb more oxygen, this is the reason why, is because there is more surface area for the oxygen to enter. Okay, it is small, but because there's a lot of surface area or a lot of contact points, okay, for the oxygen to enter. Now this one, we will learn more about it when you go to upper sec, okay, your upper secondary, sec 3, sec 4, if you take biology, right? There is a specific property, which is this surface area to volume ratio, which actually helps to increase oxygen uptake, okay, by the red blood cell. Okay, it is also elastic, and it enables the cells to squeeze through tiny blood capillaries. Now, blood capillaries are actually something very small in diameter. Okay, they are very small in diameter. That means your red blood cell really has to squeeze through it because it's very, 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 very small. Okay, this, so by allowing by the red blood cell being elastic, it allows it to go and squeeze through these small blood capillaries. Okay, now we move on to your white blood cells or your leukocytes. Okay, so leukocytes are just basically another word for your white blood cells. So they are larger than your red blood cells. Okay, they are fewer in number. Remember that your red blood cells is 99%. So this is approximately 1% of the cells, right, in blood. Okay, they are also produced in the bone marrow, which I mentioned before, which is found in the spine, and they are also destroyed at the spleen after they are being used. Okay, after they have already carried out their functions, they can no longer be used. They are destroyed at the spleen as well. Now, compared to the red blood cell, your white blood cell has very limited lifespan of only a few days. Okay, or even though they have a nucleus, so a few days, this can be from just 2 to 3 days, or even up to maybe just a few weeks. Okay, so it's not as long as your red blood cell, which can last around 4 months lah. Okay, 120 days. Okay, they are colorless. Okay, as they do not contain hemoglobin. Remember, red blood cells is red because of hemoglobin. Okay, they are irregular in shape and contain a nucleus each. They are mobile. They are able to move and change their shape and squeeze through the walls of the thinnest blood capillaries. So this is not as important. Okay, so don't worry too much about it. Okay, then of course we have your different types of white blood cells. You have your lymphocytes and your phagocytes. So what's the difference? So basically this is just outlining the different types of white blood cells. Okay, your lymphocytes are large rounded nucleus. They have small amount of non-granular cytoplasm and produce antibodies against microorganisms. So your lymphocytes, the main role is to produce antibodies. So remember I said earlier, antibodies are proteins which we use to combat microorganisms that have invaded our body, right? So your non-granular cytoplasm, all this is not important for you to know so much. Okay. So this is how it look. This is how it look. Now phagocytes have lobe nucleus, granular cytoplasm, and also to ingest foreign particles. Okay. Now ingesting foreign particles is via this process called phagocytosis. Okay. Basically, this is the process in which phagocytes surround and engulf. Engulf meaning they consume, right? They surround and engulf these pathogens, right? Or your foreign microorganisms. Okay. So how do they combat microorganisms? Okay. For lymphocytes, they just produce antibodies. They don't go through phagocytosis, right? They just produce antibodies. But your phagocytes, they will actually have a more direct effect. Okay, they are going to go through this process of phagocytosis to surround, engulf, and kill these foreign microorganisms. Okay, so this is how they will look like. Then next we have your blood platelets or thrombocytes. Okay, so your thrombocytes are basically a name for your blood platelets. Okay, again, they are not true cells, as I've mentioned earlier. Okay, they are fragments of cytoplasm which are membrane bound, and they are important for blood clotting. Okay, so for the purposes of this module, you just need to know what's the function of blood platelets. Okay, you don't really need to know exactly what they are. Great. With that in mind, okay, we'll now go through what blood group typing is. Okay, so blood group typing basically is how do we recognize different blood types, right? Okay, we all know, okay, I think it's quite common knowledge that we have different blood types, right? Everybody has different blood types. So we have your A, B, AB, and O blood group. So these are your four blood types that we have. Oops, not bloody type, blood types. Okay, so blood group typing is basically how do we recognize, okay, these four blood types, and what is the use of being able to recognize these different blood groups. Okay, there's a video here that I want you to watch, but I will not show you here. Okay, you can pause this video and you can enter the YouTube link that I've provided at the bottom here and watch it on your own accord, okay, on your own time. Then you can continue on with this lesson. Okay. Oops. Right. Now, what are so how do we, when we talk about blood groups, right, we have to talk about the concept of antigens and antibodies. Now, remember earlier on I said that red blood cells have proteins called have surface proteins, right, on their surface. Okay, so one of the proteins that we are going to talk about, okay, that is very relevant for this topic is going to be antigens. Okay, so antigens are just surface proteins in which are being produced and expressed on the surface of these red blood cells, and from there we can use them as identifying markers. Okay, why do we call them identifying markers? Because from the type of protein that is being expressed on the surface of these red blood cells, we can finally identify what blood group they are. Okay, and as you can see from these four different red blood cells, all of them have different surface markers, and even one of them has no surface markers. So you see from the different surface proteins, we can actually recognize what type of red blood cells they are. Okay, and in other words, what blood group they are as well. Okay, so that is the concept of antigens. Okay, identifying markers, identifying surface protein markers, which we can use to recognize, okay, to recognize your blood groups. Okay, now that's for your red blood cells. Okay, now your plasma, on the other hand, are going to contain natural antibodies which recognize and bind to specific antigens on the red blood cells. Okay, so you can either have an antibody A or antibody B. Okay, I will go through with you what do we mean by antibody A and antibody B and what exactly do they recognize. Okay, but for now, you just need to know that there are two types of antibodies that are found inside your plasma. Okay, and these are your antibodies A and antibody B. Now, when you want to talk about the ABO blood groups, okay, this is the ABO system. Okay, when we use ABO, this is the system in which we use to recognize your different blood groups, right? So there are four different blood groups that we know of. As I've mentioned earlier, we have your A, your B, your AB, as well as your O. Now, for a person who has blood group A, right? How do we know it's blood group A? How can we tell from the red blood cell itself for someone who has blood group A? Okay, when we look at the red blood cell, what we are going to see is on the red blood cell is we're going to find this thing called antigen A, right? So for blood group A, we are going to have antigen A as the surface marker, right? As the surface marker that is found on blood group A, right? However, for the antibody, we are going to have antibody B. It is actually opposite, right? So what is found on the antigen of blood group A people, okay, or the antigen that is found on the red blood cell for blood group A is going to be antigen A, but antibody is going to be opposite, right? Is opposite actually. It is antibody B. Okay, now again, I'm not going to go through yet. Why do we have the antibodies there, and why is the antibody opposite from the antigen? You must be thinking, I am blood group A. How come I don't have antibody A? Okay, so this one, we will just keep it there first. Okay, and I'll explain later. So if we know that on the right blood cell for blood group A, we are going to have antigen A, that would mean on the red blood cell of blood group B, on the other hand, we are going to have antigen. You have guessed it. It's going to be antigen B instead, right? So for antigen on the red blood cell for blood group B is going to be antigen B, right? And if you can see a pattern here, whatever antigen I have is going to be the opposite antibody. So for blood group B, I have antibody A, opposite as again, right? Now, here, you should be able to guess it again now for blood group AB. The antigen that I can find, right? The antigen that I can find on my red blood cell, if it's AB, that means I can have both antigens A and B that can be found on my red blood cell. Okay. However, this is the interesting part. The antibodies for blood group AB, I do not have any antibodies, no antibodies whatsoever. Okay, and there's a reason for this. Okay, and we will go through that later on. Okay, and finally, blood group O, you can tell if I can have both antigens on one red blood cell, that means for blood group O, I have no antigens. I do not have either of the antigens A or B on my surface. This is for people who have blood group O. Okay. And the interesting part again, for blood group O, I have both antibodies. I have no antigens. Okay. Again, there's a reason for this, and I will go through that next. Okay. So this is just the first part of the how do we identify the different blood groups first. Okay. It's important for you to know that blood group A is antigen A, blood group B has antigen B, blood group AB has antigen, both antigens A and B, blood group O has no antigens. Okay, and when we look at the antibodies, blood group A has antibody B, which is the opposite. Blood group B has antibody A, which is the opposite as well. Blood group AB has no antibodies, and blood group O has antibodies A and B. Okay, please take note also, right, where these things are found. Antigen is found on the red blood cell, and the antibody is found in your plasma. Okay. Now, what happens when different blood groups are mixed? So this is where we finally talk about your antibodies. Okay. Now, let's imagine we take someone who has type B blood. So we have, you should know, antigen B that is found on the surface of your red blood cells for blood type B. deeper, right? However, our antibody is going to be antibody A, right? We are going to have antibody A. Remember, it's opposite, right, in the plasma. Now, what happens when we get type A blood that is being added? Okay, what we are going to see now, remember that type A blood has antigen A and we have antibody B, right? Okay, now because type B blood, okay, let's take away the red blood cells for the type B first. Okay, but our type B blood has antibody A in the plasma. Okay, and your type A blood has antigen A. So what will happen is antibody A will bind to this antigen A. So remember one concept here. Okay, antibodies will bind to their respective, okay, will bind to their respective antigens. Okay, so what does this mean? Antibody A will bind to antigen A. Okay, so example here, like the one you see on top, antibody A binds to antigen A. Okay, so that's why you see here type B blood, right? Antibody A is going to bind to antigen A, which is on the type A blood. Okay, there you go. It's going to bind. Okay, now when this happens, okay, antibody A in plasma of recipient binds to antigen A on red blood cells of donor's blood. Okay, this will lead to an effect, what we call agglutination or clumping. Okay, so your normal red blood cells actually by right supposed to look like this, but if agglutination or clumping happens, it's going to look like this. Okay, under a microscope. Okay, now you can see here, this is just they just zoom in for you, right? How it's going to look like when the antibodies, okay, just now the example they give, right? So this should be antibody A, okay, and this is antigen A, right? For example, okay, so that is basically the role of agglutination inside your blood, right? So during a blood transfusion, the effect of the recipient's plasma is on the donor's red blood cells. Okay, because this is why only certain blood types can accept certain blood, and some can accept all, some can only accept some cannot accept can only accept one type. Okay, so when we take a look here, okay, these are the different donors and recipients that we can um, think of. Okay, so let's take a look here. Now, if the recipient, the person who is receiving the blood, okay, from the donor, which is this part, which is the top here, okay. So if blood group A donates to blood group A, okay, both of them have antibody B. Can antibody B bind? Okay, can antibody B bind to antigen A? The answer is no. Okay, of course the answer is no. It cannot bind, and because it cannot bind, right, no agglutination will occur, and hence I can donate. It's a tick here. Okay, so can A donate blood to A? Yes. Okay, now so can B donate to A now? Remember, B has antigen B, and remember that blood group A has antibody B. So if antibody B binds to antigen B, there will be clumping. And if there is clumping, that means you are not compatible, right? And hence, B cannot donate to A, right? Now, can AB donate to B? Cannot also. Why? Because AB has the antigen B and A has antibody B. So again, there will be clumping. So cannot, right? And finally, O. Can blood group O donate to A? The answer is yes. Why? Because it has no antigens. There are no antigens on the surface. And because there are no antigens, hence you are not going to get your clumping. And hence you can donate, right? And this will then be, this concept is then true, okay, for all the different donation. Okay, that's why you will see all the different answers here. Okay, sorry, right? So this concept is just used for the same. Okay, it shouldn't you just have to apply the concept of, can my antibody bind to an antigen? If my antibody can bind, that means clumping, cannot donate. If my antibody cannot bind to an antigen, no clumping, can donate. Okay, so just remember the concept at the bottom here. Okay, we are going to do the experiment for this blood group typing processes, okay, in week 3. Okay, so you can see it with your own eyes, the effect of the different blood groups on other blood groups, okay, when you try to donate and when you try to mix the blood groups together. Okay, now how is blood group typing relevant for biomedical sciences? Okay, now remember that your blood is a very essential part of your body, and sometimes when you get injured you lose a lot of blood. Okay, so when we know what blood type a patient needs who has lost a lot of blood, okay, we can then safely transfuse blood. Okay, basically we can give them additional blood. Okay, giving additional blood to patients who need it. Okay, for blood loss patients. Okay, because even though your body can make new red blood cells, sometimes if you lose too much blood, right, if you lose too much blood at one go, right, it is very bad for you and it can actually lead to your death. Okay, so to make sure that because your red blood cells are being made very slowly, okay, and only at very specific times, to make sure that you can survive long enough for your red blood cells for your bone marrow to make new red blood cells, okay, we give you the additional blood first. Okay, and this usually comes from blood donors. Okay, so this is the first part of your assignment. Okay, is that you go and scan this QR code to access the palette and answer the question. So what is the question? Okay, is can other than blood transfusions, what other ways do you think blood typing can be useful? Okay, whether it is in biomedical sciences or any part in our lives, okay, in our daily lives. Why do you think, how do you think blood group typing can be very useful? Okay, you can scan your QR code here, or and input your answer into the Padlet, or you can access the link as provided here. Okay. And finally, right, you are going to, this is for you to just test yourself, right? So what blood group does X, Y, or Z have? Okay, so just go through this. Okay, this is just an example of a type of question that they can be asked, okay, for you to determine. Okay, if you know your blood group typing concepts very well, you should be able to determine what the blood group of person X, person Y, and person Z is. Okay, this one is for you to try and test yourself, and I will reveal the answer, okay, when we meet up next. Okay, now just one thing for you to take note, serum here, right, is just basically plasma. It's another word for plasma. Okay, but it's basically plasma that is without the blood clotting factors, but that's not important. Just you can just think of it as another word for plasma. Okay, Oops. Okay. Now, you have one more assignment, okay, which is to complete the quizzes assignment below. Okay, there's the link there for you to access. Okay, this assignment is compulsory for you to do it by 10 p.m. Okay, basically it's just a very short quiz, okay, of around 15 to 20 questions. Okay, it encompasses all the concepts that were being taught today. Okay, so the deadline here is the 18th of July, which is basically the day of the release of this video, okay, at 10 p.m. So you have to finish the quiz by 10 p.m. Okay, so please just take your time to do the quiz. It's just for you to reconsolidate, okay, or to consolidate your learning from this lesson. Okay, thank you very much for going through this lesson. So please remember to complete the assignments by 10 p.m. And I'm going to see you next for our next recorded lesson next week. Okay, happy learning and see you soon.